Students, today we will discuss about the name reactions of the topic aromatic electrophilic substitution reaction. And the first name reaction in the sequence is Fries rearrangement reaction. So, what is this Fries rearrangement reaction? Fries rearrangement is basically a reaction of phenolic ester in which this phenolic ester on reaction with AlCl3, that is Lewis acid, will get converted into either ortho substituted or para substituted phenol. This particular phenolic ester will rearrange itself, means this group, that is COCS3 group, will be rearranged either at this ortho position or at this para position. So this particular reaction is called as the Fries rearrangement. What is the important point about this reaction? The first, it is catalyzed by a Lewis acid, mostly with AlCl3. Second, the ratio of ortho or para product formation, means which product will be formed in dominant amount. Either you will get the ortho product or you will get the para product. That will be dependent upon the temperature you have taken for the reaction. If you are taking high temperature, that is above 160 or 170 degrees Celsius, at that time the major portion or the dominant product will be the ortho product, that is the ortho isomer. This is basically a thermodynamically controlled product and it will be stable at high temperature. So at high temperature you will always get thermodynamically stable product, and that product is your ortho isomer. When the temperature is less, means it will be below 80 degrees Celsius, at that time para product will be a dominant product, and this product will be kinetically controlled product. So this is very important point about Fries rearrangement reaction. Next important point, if I am talking about this particular reaction, is that this reaction can have both type of mechanisms, that is intramolecular as well as intermolecular. There are several evidences that indicate that mechanism will proceed in intramolecular manner, and similarly there are some evidences that prove that it happen in intermolecular way. Next important point about the reaction is that reaction can also be carried out in presence of light. So these are the important points about the Fries rearrangement. So now we will discuss about the mechanism of Fries rearrangement reaction. So before that, I want to tell you that how this phenolic ester can be synthesized and how this is a method that can be used for conversion of this particular phenol into these two different kind of the products, these two different isomers, right? So first of all, phenol is allowed to react with the acetic anhydride in presence of sodium acetate, and this phenol will get converted into phenolic ester, and this phenolic ester on further reaction with this particular AlCl3, that is Lewis acid, will get converted into ortho or the para isomer. Now the mechanism of the reaction. This is a very important point, that the mechanism. So we will discuss the mechanism one by one. First we will discuss the mechanism, the intramolecular mechanism, and this is the intramolecular mechanism. And what happened in this intramolecular mechanism, you can see in the screen that this is your phenolic ester and this is your Lewis acid. So first of all, the oxygen with its lone pair of electron will interact with AlCl3, and there is a coordination between this Lewis acid and phenolic oxygen, this one, right? And after formation of this one, this particular oxygen is carrying a positive charge and this aluminium is carrying the negative charge. Now from here, reaction can happen at ortho position and reaction can also happen at para position. That will depend upon the temperature of the reaction we have taken. For example, at ortho attack, this particular bond will shift to this carbonyl carbon, and when it is shifted to this particular carbonyl carbon, the bond between this oxygen and carbon will migrate towards this oxygen bearing the positive charge, right? And the oxygen is also carrying the lone pair of electrons. So this lone pair of electron, while attacking of this double bond on this particular carbonyl carbon, this will shifted to form carbon oxygen double bond, right? So this particular intermediate will be synthesized in that case, and finally the H positive will be eliminated out from this one, from here, with migration of bond from C double bond O to oxygen, and here you will get this product, and this is stabilized, reason being it can coordinate with AlCl3, and finally this on reaction with ROH will accept H positive from this one and will get converted into the ortho isomer, right? Similarly, we can also obtain the para product, and in case of para product, there is shifting of lone pair from this oxygen to form C double bond O positive, with migration of this electron to this one to form a double bond between these two carbon, and this double bond will shift to this particular carbonyl carbon with migration of this bond to O positive, right? So similarly, there will be a para attack, and this para attack, after attack on this particular position, the H positive will be eliminated out from this one, right? And finally it will accept the H positive and will get converted into the para isomer. So this is the beauty of the reaction, that this particular Fries rearrangement reaction can proceed in intramolecular way. So this mechanism is the intramolecular one. Now we will come to the intermolecular mechanism, and for the formation of the for the particular intermolecular mechanism, there will be a separation. You can see that in the previous slide, you can see that there is no separation of CH3 C double bond O. Acylium cation was not synthesized in any case, but in this intermolecular type of mechanism, you are getting this acylium cation. How you are getting it? That first of all, AlCl3 will coordinate with the oxygen with the same mechanism as happen previously, and after this one CH3 CO positive will be separated out. So acylium cation will be synthesized, and to this acylium cation there will be ortho attack or there will be para attack depending upon the reaction condition that we have taken. So here this is particularly ortho attack, that this particular attack at this position, and para attack is that this bond will shift here, then this bond will migrate to C positive and will form again this type of the intermediate, and after formation of this one the H positive will be eliminated out, which finally abstract H positive from the solvent you have taken, and it will get converted into OH, that is either ortho or the para product. So this is the reaction which proceed with intermolecular mechanism. So the major difference between the intramolecular mechanism and the intermolecular mechanism is the separation of the acylium cation in the case. The acylium cation has been separated in case of intermolecular mechanism, whereas that have been not separated in case of intramolecular mechanism. Instead, there will be a directly attack of the double bond to the carbonyl carbon, and then it will be separated from the complex. Now, the important point about this reaction that I was talking about, the evidence of the Fries rearrangement, that it can proceed in intermolecular as well as intramolecular mechanism, that this is a evidence about the reaction in which we have taken a mixture, and on this mixture we find out that this particular carbon is isotopically labeled, right? And after identification of the product, what we found that we got these two type of the products, and other products were also be synthesized. So if we are getting this particular type of product, that is the isotopically labeled carbon is being a part of this particular product, means the mechanism is an intermolecular one. Similarly, there are some evidences that prove that the mechanism is intramolecular in its nature. Now, we were talking about a point that the Fries rearrangement can also proceed in presence of light, that is called as the photo-Fries rearrangement reaction. And what is this photo-Fries rearrangement reaction? That is conversion of this phenolic ester into the ortho or the para isomer in presence of sunlight and ethanol, right? So here is the formation of these two kind of the products. The beauty of the reaction is that besides these two products, we are also getting a very minor product, that is phenol, in the photo-Fries rearrangement reaction. What will be the mechanism for the reaction? That is also important. In case of photo-Fries rearrangement, the mechanism will be free radical type, and there will be a formation of free radical initially. Acylium free radical will be separated out, and there will be a formation of this one, that is O radical will be here, right? So there will be an attack of this particular double bond, that is a single electron will attack on this particular radical, and either ortho or the para attack will be possible depending upon the condition, right? And how we are getting a minor product, that is alcohol, that can be synthesized, that is we are getting the phenol. Phenol can be obtained, that this particular your free radical will abstract H radical from the solvent and will get converted into a very minor phenol product. So this is the beauty of the reaction, that we are getting a phenol as a minor product in the case of reaction. So this is all about the Fries rearrangement, that is conversion of phenolic ester into ortho and para substituted phenol by using intermolecular, intramolecular or photo-Fries rearrangement type of reaction, right? Now the next reaction. This is also a very important reaction, that is called as the Reamer-Tiemann reaction. And what makes this reaction important? That is formylation. Formylation of this particular phenol. This is your phenol, and this phenol on reaction with chloroform in presence of base. For example, we have taken potassium hydroxide here at a temperature range of 60 to 70 degrees Celsius, will get converted into CHO group. Means at ortho to this hydroxyl group there will be a attachment of CHO group, that is called as the formylation reaction. Means basically the formylation of phenol with chloroform in presence of a base is called as the Reamer-Tiemann reaction. Important point about the reaction is that there are formation of two products. We can also get orthoisomer and we can also get para-isomer, but the major product is orthoisomer as comparison to para-isomer, right? Reaction proceed with the formation of carbene, that is dichlorocarbene. So this is the beauty of the reaction, that we can insert CHO group to the phenol at a position ortho with respect to the hydroxyl group in the major proportion, right? That can also be inserted at the para position, but that will be always, in the minor proportion. Ortho proportion will be always the major, right? So mechanism of the reaction, if we are particularly talking about the reaction mechanism, will start with the formation of dichlorocarbene and attack of dichlorocarbene, reaction of the particular phenoxide ion, because the reaction is taking place in presence of base. So phenol will get converted into phenoxide ion. So phenoxide ion will react with the dichlorocarbene, that is your intermediate in the reaction, and finally you will insert the CHO group ortho to the hydroxyl group into a benzene. This is the mechanism of the reaction. If we are particularly talking about the mechanism, first of all, I was telling you that this base will abstract H positive from this one, and this will form CCl3 negative, which finally loses the Cl negative and will get converted into dichlorocarbene. After this one, the phenol will react with the base and will get converted into phenoxide ion, and phenoxide ion, this O negative, this electron pair will shift to form C double bond O, and this bond, that is the bond between this carbon and this carbon, will shift to dichlorocarbene, right? And this will form this type of complex. After that, the C negative will attack on H with migration of electron to form again C double bond C and O negative. So there is formation of CH-Cl2. Again we are reacting it with base, so OH negative, that is a nucleophile, will now attack on this particular carbon. OH negative will attack on this one. So there is the attack of OH negative with migration of Cl. Means how this will occur, that this is your OH negative, and this OH negative will attack on this carbon with migration of one Cl. When Cl is migrated, it will form this one OH and Cl, and then finally this O negative will abstract H from the OH with migration of the bond between hydrogen and oxygen to form C double bond O with elimination of Cl negative from the system, and we are getting here insertion of this C double bond OH group ortho to the hydroxyl group. So this is the mechanism of the reaction, that how the reaction can proceed and can convert into salicylaldehyde. So this is the beauty of the reaction. Now this reaction is synthetically very important, and importance is that beside phenol, some activated heterocyclic compounds can also react with chloroform in presence of base. This is the beauty of this reaction, that how they can be converted into this one. For example, if we are taking the example of pyrrole, right? So if we are taking the example of pyrrole, I am particularly talking about this is your pyrrole, this one, and when this is allowed to react with this reagent, what will happen here? This pyrrole will converted into this, right? So CHO group will be attached to the second position with respect to the nitrogen. So this is how the heterocyclic rings can also be formulated by using chloroform in presence of base. Again, the mechanism will be same as in case of phenol. It will start with the formation of dichlorocarbene, then attack of this double bond to that dichlorocarbene, and then finally the attack of the hydroxyl group, and with elimination of Cl negative from the system. Second application of this reaction, that is, this is the first application of the reaction, that the activated heterocyclic ring can also be formulated by using this particular reaction. Second application of this reaction, if I am particularly talking about, is that instead of chloroform CHCl3, if we have taken CCl4, carbon tetrachloride, that what will happen now? Then there will be no formylation. Instead, there will be introduction of carboxylic acid group ortho to the hydroxyl position, right? At that time, we will get this type of product. Means if we have taken carbon tetrachloride instead of CHCl3, at that time salicylic acid will be synthesized and we will not get salicylaldehyde as the product. So this is the beauty of the reaction, that how it can be used for the preparation of the heterocyclic compounds that can be formulated, and preparation of salicylaldehyde and salicylic acid from phenol. So that's all about today's lecture. Tomorrow we will discuss some more reaction of that is related with the aromatic electrophilic substitution reaction.